Hi, everyone. Welcome to our Wednesday session, webinar session, actually. So today we've had a few of these in the last few weeks, which is wonderful, and we've had quite a good response. Today we're doing grapevine trunk diseases, causes, impacts, and management techniques with Dr. José Ramón Úrbez Torres from the Summerland Research Center in BC. To start off, I will have this QR code for the CEU points. So I have done some research, and this is for the CEU points. I am trying to figure out what is going on with the pesticide points as well. So that's where some of the confusion came in. But I would allow Dr. Torres to introduce himself and take it away. Thank you very much.
Okay, thank you. I'm gonna put my presentation first.
Ooh. There we go.
Let's go through all these. Display. Okay, it should be good now. Can you see it? Good?
Yep.
Okay, let's see my pointer. Good. Well, good afternoon there. It's 9 in the morning here in BC. And yeah, thank you very much for the invitation. Thank you very much, Katarina. I appreciate the opportunity to share some of my research here in BC, but also to give general information, talk about an important problem, you know, on vineyards, not only in Canada, but around the world. I am a research scientist at the Summerland Agriculture and Food Canada. I'm a plant pathologist. My expertise is on perennial woody crops, primary grapevines, but I also do research on tree fruits, mostly cherries and apple here in the Okanagan Valley. My background, I'm originally from Spain, so you will have to keep up with my Spanglish. Sorry for that. But I moved almost 20 years ago. I moved to California, where I completed my PhD in plant pathology at the University of California in Davis. And in 2010, I moved here to BC as a postdoctoral researcher and was able to compete for this position in 2013. So since 2013, I've been working here for the federal government in Summerland. And as I said, my program focus on, you know, conducting both apply and fundamental research on all aspects of plant diseases, you know, from identification, developing diagnostic tools, understand epidemiology, biology, and trying to put all this together to provide management solutions to the industry here in Canada. So that is in a nutshell, and it's a little bit of what I'm going to be talking today. So if we start our presentation, as an outline here, I will give you a brief introduction, you know, what grapevine trunk diseases are, why they are so important. I will give you a snapshot of the research that we have done in British Columbia and where we are at. And very important to continue our presentation, I will talk about the life cycle epidemiology and the inoculum sources, because this is going to be key, you know, for the following steps we are going to talk to understand management strategies. I will talk about a very important part, which is the status of these fungi on the nursery material or the planting material, and you will understand why and the implications they have for growers or for the wine industry. I will touch in a very important point, which is understanding the epidemiology, and this is going to be key, you know, to really implement proper control methods, which I will be talking about chemical practices, biological control, but also cultural practices we can put in place to mitigate these diseases. So I'm pretty sure most of you, if you are a grower and you grow grapes, you are familiar with grapevine trunk diseases. You have seen these symptoms before. But very briefly, the grapevine trunk diseases are caused by many different fungi, and this is one of the challenges, the biggest challenge we have with this complex of diseases. This is not powdery mildew, where we have one disease, one pathogen, or botrytis, we have one disease, one pathogen. Currently, there are about over 100 different fungi that can cause grapevine trunk diseases, and if you think that about management strategies, it's very difficult to find a silver bullet, like a biological, a chemical that is going to have such a wide spectrum to control all these 100 different fungi. So that's one of the biggest challenges we have with these diseases. Another important thing is these pathogens infect grapes through wounds and openings. And as you know, every single year we need to prune our vines if we want to get a crop. So we are actually favoring the entrance of these pathogens in our vines. Any other type of wounding, like mechanical damage, retraining, it can actually be a point of entrance for the spores in the plants. We know now that some of these fungi are thought to be latent pathogens, and I will touch that later when I talk about the situation of these fungi in the nursery. Latent pathogen meaning they can be part of the plant, they can be part of the microbiota of the plant, and is when something happens in the plant, some factor, whether it's a stress, triggers, and these fungi go from latent pathogens to virulent. And I will talk a bit about that. The overall symptoms, depending, the disease you have, depending the complex you have, depending the fungi you have in the plant, because as I said, there are over 100 and not only one is found. Sometimes, many of these fungi can be found in a single plant. We observe a slow or rapid dieback or even a sudden vine collapse. We can see mostly a sudden progressive death of the plant parts, as I show you here, so the fungi start infecting. So we can see here a cordon completely infected and dead, while the rest of the plant is still intact and healthy. And internally, we see mostly these perennial cankers. This is the area, the vascular system is compromised, is completely killed by the fungus. And of course, there is no translocation of nutrients of water to the top. And as you can imagine, as this symptom progresses, it will cause eventually the death either of the cordon, the trunk, of the entire plant. The distribution is all around the world. There is no place that we say we are free of trunk diseases, but it's true that depending the geographical region, we are going to have incidence of some pathogens than others. So we can divide trunk diseases in two major groups, those affecting young vineyards, and a young vineyard, depending where we are located, can be 5 years old or can be still 8 years old. So, but usually, within the 5-6 years after planting, we consider a young vineyard. We have two main issues, which is blackfoot and Petri disease. Blackfoot is the only disease here where the causal agents, the fungi in these different genus, are soil-borne, and usually blackfoot affect the root system. That's why it's quite an issue in young plantings. Petri disease is also issue in young vines, and it's caused from these three main group of pathogens, and altogether they form what we know as the young vine decline. As symptoms, we can see again, the necrosis, the fungi compromising the vascular system in young plants, and we are going to observe, this poor vigor. Sometimes, these vines will not even produce any spring growth, when the season starts, and eventually will collapse. In mature vineyards, mostly we have what we call the canker diseases, which is Botryopsphera, Eutypal, Phomopsis dieback, and I just saw you already this typical canker. But also we have ESCA. ESCA is probably one of the oldest diseases that we know to occur on grapes. It's still poorly understood, believe it or not, with almost over 100 years that we know that this disease is happening, but it's a very, very complex disease. And the main difference in this disease is also includes some basidiomyces. These are the group of fungi, mushroom type. As you can see sometimes, when you walk in the forest, these mushroom caps. So the basidiomyces are different from all the rest of the fungi with our ascomyces, and they cause this typical characteristic yellow spongy symptom in the bud. Even if you press and you touch, it's very, very soft. So why do we care about grapevine trunk diseases? Well, it's considered right now the main biotic factor limiting the yield and also the lifespan of our vineyards. These are economic studies done in California, and here we can see as the vineyard age, the incidence of trunk disease increase. So we can see that we are start seeing maybe symptoms in year 7, 8, 9, And for example, we are even year 12 or 13, we can get even a 50% incidence in our vineyard. As the age progresses, the incidence increase. And as the incidence increase, our productivity decrease. As the vineyard age and we start seeing the symptoms, we start losing parts, spurs, cordons, we start losing vines. So studies in California have shown that even, in year 10 or year 12, heavily infected blocks for trunk disease can lose up to 20% of production. By year 15, this case was done in the San Joaquin County in Central California. We can go even 50% production. So as you can imagine, in a very short time, our vineyard is not going to be economically unsustainable anymore. But this is not only in America, it's a problem worldwide. And this is just some data, of the situation of trunk diseases. In France, ESCA is the major problem. About over 10% of the vineyards in France are affected, and they are estimated that they are not economically viable. About 100,000 hectares, which is the loss of almost 1 billion euros. And this is some data coming from France in just one disease, ESCA. In California, there are studies that growers spend more than 260 million a year just to try to control and mitigate the impact of these diseases. And just one disease in one cultivar, you type a dieback in the main cultivar in Australia, Syrah, can cause losses up to almost $3,000 per hectare. So quite significant, the damage and the impact that this disease causes in the productivity. And most important, these diseases are slow progressing. So we are seeing, the dieback and declining over the years. And there is a point where, of course, our vineyards are not going to be economically viable. In terms of the situation of grapevine trunk diseases here in Canada, they are not new, and actually some of the first reports of trunk diseases came in here in North America in the early 1900s. What we knew it was called dead arm disease. The first report in Canada, we have it in the mid-60s in Ontario, and there have been some studies in Ontario and Quebec, but these studies have been mostly as part of big surveys conducted in the United States, in the Northeast United States, and they have actually reported some of these species, but they were not properly in-depth studies in these regions. Most of what we know on grapevine trunk diseases in Canada is the work that we have been conducted here in British Columbia in our laboratory since 2010, when it's basically when the industry started realizing the problem and targeted trunk disease as a priority. So field surveys conducted, we have about 95% of the vineyards. We have vines with symptoms of trunk diseases. We did a survey where we monitored over 70,000 vines, over 2 years, and we estimated that about 10% of those vines were showing symptoms. These were the vines that were only showing symptoms. We know there can be infection with no symptoms appearing. And right now we have over 40 different fungi that we know cause trunk diseases here in British Columbia. So also, this disease, as I mentioned before, progresses at the age of the vineyards. So we can have here all our vineyard blocks in that survey and the different ages of these blocks. We can see here a big incidence in some of the young blocks, and this is caused by the young vine decline I was mentioning before. We have some issues with some vineyards within the first 5 years where, because of this blackfoot petri disease, young vine decline, we have a severe impact. Usually we have this kind of area between 10-15 years with not high incidence, but this is because that's when we start really working in our vines. We start really doing the much more heavily pruning. We start forming our condors or maybe if we are cane prune. So that's where we start seeing the infection happening. The infection starts usually, year 5, 7, 8, when we start doing more pruning cuts in our vines. And it takes time to see the symptoms. It's not like the spore lands in the pruning wound and the same year we're going to see the symptoms. Some of these diseases, we don't see symptoms until maybe 3 or 4 years after infection. And that's what is usually happening. When the blocks start getting older, we are seeing all the infections and symptoms really going higher up. Unfortunately, in 2024, as many of you know, we experienced a very deep freeze here in January. We went to minus 28 degrees for almost 3 days with, the highest of minus 18, minus 19. And this caused a significant damage to our blocks here in BC. I took the opportunity of that situation to conduct a field survey because a lot of blocks were going to be retrained or going to be pulled out. So I was able to get huge support from the industry to run this survey. Very simple. We went through almost 159 blocks, red and white cultivars planted between 1990 and 2021. We have 10 vines per block, and basically what we did is selecting 2 panels in the middle of the block, and in the 10 vines, in the 5 vines per panel, 10 vines in total, were cut about 10 to 15 centimeters above the ground. And we were taking pictures, digitalizing all this. And as you can see here, we were measuring, the percentage of the canker in the area, in the total area of the section. This is one block of the 10 vines, and you can see here that every single vine of those 10 have the canker all the way to the bottom of the trunk. So 100% of the vines were infected. 100% of these vines we cut have the canker at the very bottom, which was not a very good thing to see. But if we take into consideration that we don't have really too many control products still here in Canada, some of these blocks were significantly old. But the message is that the infections were making it all the way down, in most of the blocks. So some of the data here, again, those blocks planted, and older, between 30, 35 years old have the highest average percentage at the bottom. And you can see kind of the trend that the younger the block, the least amount of canker at the bottom. It still was quite surprising to see some of the very young blocks still having already like a 20% area in the canker at the bottom, which this is something that I haven't seen before in other regions. And I believe because the way we grow grapes in Canada and the stress conditions these vines are here in Canada, it may impact, and it may accelerate the progression of these pathogens into the vines. We have some data regarding cultivars, and you can see here that we saw a huge difference, depending the cultivar. We had to take into account that, there are some cultivars we have more blocks or they were older than others, but usually we see a similar pattern in other regions, Sauvignon Blanc, things like Cabernet Franc, Cabernet Sauvignon, they are highly susceptible. And, we saw things like Grenache, Mourvedre, or Muscat, some other cultivars that they were less susceptible. There is quite a difference in susceptibility. All Vitis vinifera, is susceptible to trunk diseases, but we know there is some difference in susceptibility. So I'm going to finish this first part just showing the grapevine trunk disease life cycle and the source of inoculum, which is going to be key for what we are going to continue in the rest of the talk. So grapevine trunk diseases can first, as I mentioned, enter in our vineyards through contaminated nursery material. And I will explain this and show some data we have right now on that. This material can be planted, introduce these pathogens and have already the disease in our vineyard. Usually what we are seeing is as vineyards are getting older, we are doing all these pruning cuts and the spores are getting our pruning wounds, infection develops. We are going to see in these affected areas the fruiting bodies, these structures where the spores are within, and they are basically waiting, resting there for the optimum environmental conditions, weather, rain, temperature, so these spores get released. Of course, the fungus is very smart and it's going to get the spores released when it's the biggest chance to reproduce and to continue the life cycle. So when this happens, so usually when we prune, so those spores are going to be in the environment, are going to infect our pruning wounds, and the life cycle continue. As you see, the cankers continue developing, symptoms again. Another challenge we have with these diseases is they have a very wide host range. It's not only happening on grapes. There is, for example, I give you Eutypa dieback. It has been described over 100 different hosts that Eutypa dieback cause disease. Here in the Okanagan, in BC, our main issue can be cherry and apple. They are also host on these pathogens, so we grow cherries and apples, block after block between vineyards. So that can be, another source of inoculum. We also know that many also native plant communities or even introduced plant communities can be hosting this fungi. We can talk from, oak, willows, even pine trees in California. We found this disease is even affecting sequoia. A lot of different bushes, etc., can be also inoculum. So it's a very difficult disease to control just by managing, the host or the surrounding area, because the hosts are very broad. So as a summary of these parts, so grapevine trend is the main biotic factor, limiting yields, cause significant economic losses. They primarily infect through pruning wounds, but can be also introduced in the vineyard with the planting material. They are widespread, and our studies in BC show that 100% of our vineyards we have been testing so far are infected. And as I said before, this doesn't mean that we are seeing symptoms above the ground, symptoms on the foliage, but this canker is inside the vines. And we have different incidents depending we have a young or mature vineyard of these diseases. So I'm going to move to the next part, which is to study the status of these diseases in nursery material and the implications they have for the wine industry. And this is something very important here in Canada. As many of you know, or probably all you know, we are not really a country that has a very large nursery industry. We do have nurseries in Canada, but most of our material comes from the 3 countries that are allowed to export grapes in Canada, which is France, Germany, and the United States. The Canadian Food Inspection Agency certifies nurseries in those countries that are allowed following the specific processes to export to Canada. We do have some production, some domestic production, but of course it's not enough to keep with the demand. And still, for example, things like the rootstock still mostly come from internationally from other countries. So we have a big, influx of material. So we really wanted to study, the status, the health status of this planting material coming into Canada. And it's not new that we know that plant disease fungi are in planting material. However, until now, most of the studies conducted primarily in Europe have focused on the presence of absence of this fungi in the material. But our theory is that maybe having this fungi in the material doesn't mean it's gonna collapse, it's gonna die. So it's really the presence of the fungus equal to plant death. And for that, what we wanted to do is really to quantify the amount of pathogen that can come in that material, because we believe that the amount of pathogen inside the plant is going to play an important role on causing disease on the plant. So for this, we studied 4 different nurseries. We collected 30 plants per nursery, all these different cultivars, and we have cell-rooted or grafted on 3309. We develop a molecular system based on the droplet digital PCR who allows us absolute quantification. This is each of these blue dots you see here is a positive for our target of a specific pathogen, and each column is a different vine. And we look in 4 different places for each plant, in the roots, at the base of the trunk, in the graft union, and in the scion for the plants that were grafted, or in the scion at the end of the trunk and the roots on the plants that they were cell-rooted. So the results we got from the different nurseries, and I can show you here, nursery A and nursery C are the same nurseries, but they are sampled in different years. These are all our different pathogens we were testing, and this is the percentage of plants infected in each nursery. And as you can see, a lot of 100%. 100% of the plants, only 1 plant in all the 150, we were not able to find any of our targets. And we have an average of 3 fungi per plant. But you can see also here quite a lot of variability, depending, the pathogen and also the cultivar. For example, here in 2014, 100% Salmonella chlamydospora in Pinot Noir, but only 30% in Chardonnay. If we move to 2018 from the same nursery, we see that we went in Pinot Noir from 100% to 50%. And this is very common. There is a big, huge variability in the amount of incidents and pathogens we found. And as you can think about it, nurseries, collect material sometimes from different mother blocks, different scion blocks, so that can be also playing a role, or even the step that they have in the nursery process. So the total fungal abundance per nursery, as I mentioned you before, so these are the different nurseries, and you can see here the difference in the fungi. In red, Phaemonella chlamydospora, heavy, high infected in nursery A in 2014, significantly reduced in 2018, and almost no present in other 3 nurseries. Very similar situation with other fungi. We have here, for example, in blue, Botrytis heavily present in the nursery A, but only in nursery D we were able to find high numbers of Botrytis. One species, Cadophora olivaceae, it was the one we found across all nurseries at high levels, but still you can see some difference. So just to show you, the huge variability we can have between the nurseries and the pathogens in those nurseries. But also we can find a significant difference and variability, if we compare the plants within the same nursery. So these are 10 plants coming from the same nursery, Phaemonella chlamydospora. And you can see in the different parts of the plants, the base, the graft union, the roots, and the scion. So for example, if we look here, plant 4 and 5, they are pretty heavily infected, for this fungi in the base, the graft union, the scion. But then we look, for example, plant 3, almost or no presence in the scion, very low in the graft union and the roots. Same in plant 9, for example. So you can see the significant difference. So that's why it's quite difficult, to really assess planting material and the significance that this can have in the nursery. So based on that, we came to this hypothesis. So if we are finding 100% of these fungi in the planting material, and we believe that having the pathogen means that plant is going to die, is going to collapse in the field. I mean, my thought is we will not have viticulture as we know these days, because we found 100% of these plants are hosting these fungi. So our hypothesis is the presence of the fungus may not result, on disease and eventual death. And just showing you, explaining this figure, this is the grapevine lifespan, and this is the fungal colonization in the plant. So if we have a vine, with its, growing healthy under no stress conditions, the colonization threshold for the mortality of this vine is going to really come at the end of the lifespan of this vine. So this vine is going to be productive for many years, because it's going to have a balance. However, if we have, for example, one pathogen and this vine is under stress, this mortality is going to come earlier. We're going to have a premature death. And our hypothesis is this mortality is going to become even earlier if we have more than one of these pathogens in the plant under these stresses. So this is the hypothesis we were managing, and why we are not seeing hundreds of the plants dying in the field. So for that, I have a PhD student, Jerry Rykan, that completed his PhD last year, is right now conducting a postdoctoral research in Australia. And we were looking into how abiotic or biotic stress factors may impact or influence the development of this disease. So we look at very different things, but also very important, we wanted to know what the inoculum thresholds in the plant means. What means having in the plant a very high amount of the pathogen or a very low amount of the pathogen, because this is what we are actually seeing coming from the nursery material. Our hypothesis being plants with a much lower amount of the pathogen are able probably to contain that pathogen, to be able to overcome. But plants with a very high amount of pathogen, the defense system is not going to be enough to contain that pathogen, and the pathogen is going to take over. So the first thing we have to do is to actually develop a system that we can, mimic the situation we are seeing in the planting material, because we have to introduce these spores inside the dormant cane. So we develop a vacuum system. So basically, this is our vacuum system. It's applied at the top of the cane, and we vacuum inoculated the spore suspension into the cane. So basically, you can see here from the base all the way to the top, we implement our molecular diagnostic tool, and we can see the vast majority of the spores get in the first 8 centimeters, so in the first node. Yeah? And this it has some logic, because then when we have the nodes, and the vascular and the silent vessels, we have some difficulties for the spores to progress. But basically, we're able to get the spores in the entire cane. So that way, we were able to infect the canes with the exact number of spores we wanted to conduct these experiments. So our first experiment was to evaluate water stress and how the deficit irrigation would impact the development of the disease and the growth of the fungus. So we have a 2 years experiment in greenhouse, and we obtained a significant water stress. And you can see here the scorching of the edge of the leaves, a very clear sign of water stress. We have actually a heat wave in July, where even the plants who were well irrigated in green here under no stress, they saw a stress. And we have also soil moisture measurements, so we have actually good separation. So we were, sure and confident that our plants were having water stress. So at the end of the experiment, of course, the plants under stress here is they have less node numbers, less pruning weights, the root weight was lower, as expected, those plants are under stress factor. But the most interesting thing is we have an increase in the fungal abundance in the plants. So those plants under water stress have a much higher fungal abundance than in the non-stress. So we inoculate those plants with a known amount of pathogen. Those were between 2,500 and 25,000 spores. And we knew that under water stress, at the end of the experiment, the amount of pathogen was higher. Also, we conducted a 3-year field experiment, very similar, 2 different levels of inoculum, non-stress, 100% water, and stress deficit irrigation of 50%. At the end of the experiment, we measured the plant mortality. So our controls, they were not inoculated. We had some mortality just on the stress vines, but very interesting. Those plants that had a lower amount of spores, the plants under stress have a much higher mortality, almost 60% of the plants die, versus the plants who were with no stress, a much lower mortality. So our hypothesis was the plant having a much less amount of spores, if it's not under stress, can contain that fungus. While if we impose stress, the fungus is going to take over. Plants who were with a very high amount of spores, inoculum, 25,000 spores, it didn't really matter if they were under stress or non-stress, because there is such amount of inoculum in that plant, the plant cannot contain and cannot defend. So the water stress definitely caused much higher mortality. So we were able somehow to prove our hypothesis. So these are some of the pictures of these vines. We have, inoculated with 25,000 spores, no water stress, 100% water. We start seeing this collapse, this reddening of the leaves. 25,000 spores under stress collapsing, 5,000 spores under 50% irrigation collapsing. Most of our vines with a low amount, 100% of water, they were looking healthy. They were looking green. So that was quite significant to observe these symptoms associated with the deficit irrigation and the amount of inoculum we had. So as a summary for this part, so grapevine trunk disease fungi are present in ready-to-plant nursery material. However, the abundance varies significantly between nurseries, between plants from the same nurseries, and even between different sections of the plant. So we have proven that water stress can increase the pathogen growth in the greenhouse and also increase mortality in the field under plants with a lower amount of inoculum. And we have a much higher mortality in plants inoculated with high inoculum. The pathogen presence may not imply plant mortality, but what we believe is these abiotic or biotic factors, stress can favor disease development. I don't have time to show here all the work we did, but we also look into the impact of nematodes as a biotic factor, and we saw that nematodes, ring nematode, work I've done here with nematologist Tom Forsch in Summerland, the ring nematode, which is quite present in our region in BC, actually influence and increase the presence of the stress that the ring nematode cause in the vines, in infected vines. We also have another experiment with heat treatment. We were mimicking what happening under heat dome, and we observed that those plants that they were highly infected, artificially inoculated under the plastic that we produced there to mimic the heat dome, we saw a much higher mortality. So, of course, the home take message for the industries, we should do everything possible to avoid the stress, of these plants during these first years of establishment, because then we can see an increase on disease. So moving quickly to our next part, I want to talk a bit about what is the epidemiology of grapevine trunk diseases and why understanding this information is so important for the management practices. So with epidemiology, what we really want to look is the presence of the inoculum in the vineyard, when it's going to be present, and when we are going to have these high or low infection periods in the vineyards, when the spores are going to be acting in our vineyards. So here, and we can also correlate what environmental factors favor this spore release, because then we can actually start working in models on a way to predict when these spores are going to be out. So we conduct here a 3-year study in the Okanagan Valley. This is the old Okanagan Valley, where we have 5 spore traps. This is Burkhardt spore traps. They work 24/7, and as you can see here, there is intake of air. The spores gets in these Eppendorf tubes that we collect weekly or daily, depending, and we monitor, of course, with weather stations. So same as I mentioned before, we developed a molecular system where we were able to collect these tubes from the field, extract total DNA, and using PCR, draw digital PCR, we were able to absolutely quantify the amount of spores in those tubes. So here, this is just one example of the Botrytis farinaceae causing bud canker, spores trapping studies in British Columbia. So we have here the columns is the total spores per week. Here we have our temperature through the 3 years, and here we have the precipitation, taking into account that, yes, of course, most of our precipitation in the winter months of December, January are usually in form of snow. So the first things we see here is that the first significant spore release happening, at the end of winter to early spring. And it's very interesting to see that not always is correlated with precipitation. So we are seeing that as soon as we are getting out of freezing temperatures, average getting to 5, 6, 7 degrees, that's where we are seeing our first spore release. And then the spore release, continue through the growing season, highest usually during the months of mid-April to mid-May, end of May, early June. We have some peaks here, and usually that's probably where the fruiting bodies are most mature, highest amount of spore release. But as we can see here, we see, most of the spores released through our growing season. Very interesting, not always correlated with precipitation. During the winter months, there are none or very low during spores present during the winter, as we can see here. So we can see basically these patterns, every single year, growing season, winter. So I will come back to this, but just want to show you how we can use this data to really use for management strategies of trunk diseases. And that's the last part of the talk, how we can control and what we can do to control grapevine trunk diseases. And I will talk a little bit about the cultural practices, chemical control, and biological control. So the control of grapevine trunk diseases, as I mentioned before early in the presentation, all Vitis vinifera is susceptible, but yes, we see differences in susceptibility. Wild Vitis species and even hybrids have been shown also to be susceptible. There was some work conducted in the States. We used to have products that they were able to stop the infection, once the canker was in the vine. Sodium arsenate in Europe was banned in 2000, or the phase out of Venomil, which is one of the systemic fungicides. So these products are no longer available in many countries. I mean, sodium arsenate was completely banned in Europe. And it's very interesting to see the explosion of grapevine trunk diseases, in the early 2000s until now. So we saw a correlation, eliminating these systemic products that could contain and control the infection inside the plants, to the major epidemics we have these days with trunk diseases. So as I mentioned before, right now we don't have a systemic product, chemical, biological, that we can treat our vines to stop the infection. So most of our control strategies are going to be controlling the pruning wounds, pruning wound treatments to avoid those spores to enter in our vines. But we also have some cultural practices, and I will mention some of them going in the presentation that we can use to minimize, to mitigate. And as I said, it's a disease that is very, very difficult to eradicate. And I would say it's almost impossible to eradicate. But I think we have right now management studies that we can mitigate to a point that we can, hopefully live with it within a vineyard. And one thing is, until very recently, we have absolutely no control product registered here in Canada to control this disease. This has changed, fortunately for us, and this is work that we conducted here in Summerland with minor juice, David Neal, and we were actually trying to register a product that we know it works, thiophanate methyl, based on work that I actually conducted in California. It has a wide, broad spectrum, so actually this product affects many different fungi causing trunk diseases. Here in Canada is Senator 50SC. And we can see here, two different rates. And this is the percentage of vines infected, 24 hours, 14 days, and 21 days. So we can see that even after 21 days, the product is very, very effective and still active on our pruning wounds. And we are significantly reducing the amount of infection in our pruning wounds compared to the non-treating controls. So thanks to this work, finally, we have the first chemical product registered in Canada against trunk diseases, Senator 50SC. And here is just the label and some of the application and rate you have to use, so you have that available. So yeah, so finally we have a product here in Canada. But I want to move to some of the work we have done here, because also our industry here in BC is demanding, alternative solutions to chemicals. Here in British Columbia, over 50% of our industry is organic. So we have a very large acreage of organic grapes growing. So we wanted also to provide solutions. And what we wanted to do is just not bring in products from, registered in other countries that we know it may work or not, but mostly looking for some locally sourced biological control products that we can find here in BC that we can hopefully prove that they have a biocontrol activity. So a master student I have in my lab, Jinx Pollard, was working on this project. And basically, the whole idea was to try to find these biological controls on the genus Trichoderma, which we know they are biological, they have a high biological control, and conduct different studies to see if we can provide some alternative solutions for this. So overall, in a summary, we were able to find 7 different species of Trichoderma here in BC, and this is just, a molecular analysis and morphological studies to identify them. We find 2 new species, Trichoderma canadense. I will talk later about that because it's quite important what we found on this species. And we conduct both in vitro laboratories and in planta, because we have over 30 different isolates. Of course, we have to nail down which are the best species and the best isolates that we can move to the field to complete experiments. Like it will be very time consuming and very, very, very hard to move 30 different isolates. So thanks to these laboratory and greenhouse studies, we were able to identify 3 species from BC that they have very, very high biological control activity when we apply them as pruning gum protectants. So of course, our next step was to conduct a field trials under environmental conditions to see how they perform in nature. And these were our 3 treatments. We have a combination of 2 species because it has been shown, and we were able to show that a combination have a higher biological activity than the species applied by themselves. So we have 2 years, and this is basically showing you the larger the bar, the higher control we are having on the pruning wounds. 2 different trunk diseases pathogens, Diploida ciliata and Fusikum parvum. Here we have our BC local isolates, and we were comparing against commercially available products in other countries. T77 is a biological control based on Trichoderma. Viticil is a polymer, also can be used in organic farming, and Gilsil is a tebuconazole-based product. So overall, what we saw is our locally found Trichoderma species had similar or even best, much better control activity in the pruning wounds than some of the commercially available products. And very important, the trichoderma was really lasting long in our pruning wounds. After 60 days, we were still reaching, with BC2 or BC3 over 90% protection of this pruning wound, which usually none of the chemicals are able to obtain. The chemicals, after 2-3 weeks, they will need to be reapplied because that activity is lost. With the biological control, we are seeing a much longer control activity in that pruning wound. I come back to the species I mentioned before, Trichoderma canadense, and one of the things we found, we were doing a spore germination. As you know, this is a biological agent. It's a fungi that has to be put in the field, and it has to have optimum conditions for these spores to germinate in the pruning wound, colonize the pruning wound, and have that pruning wound protected before the pathogen comes. So what we found with Trichoderma canadense was the only species who had actually very high germination rate at lower temperatures. And when I talk about lower temperatures, we can see here at 15 degrees, even 24 hours later, we were reaching 90% germination of the spores. Even at 10 degrees, after 24 hours, we started to see germination. None of the other Trichoderma species were able to germinate at these lower temperatures. And this may be something that can favor us in cool climate regions, because we are going to need to put these products when temperatures are really not reaching the optimum, of these biological controls. Here in BC, for example, if we are looking to start protecting pruning wounds in March, we are going to have average temperatures between 5-10 degrees. So this species looks like it's more adapted under these temperatures. So that's why it's quite significant, this finding. So just to show you the significance of this work, it was awarded as the best viticulture research, by the American Society of Vineyard Viticulture, and that tells you, the impact and the significance of the work we are conducting here in Canada to the international community. We have right now a very high potential biological control locally found here in Canada that can be developed into a product. So the other thing I wanted to talk, we have talked about chemical control, we have talked about biological control, and I'm going to finish talking about cultural practices, things we can do in the vineyard to mitigate. And one of the things we know we can do is managing the pruning. This is work I conducted in California while I was doing my PhD. Again, epidemiology, spore trapping. We can see here in California when the spores of Botrytis happening. Mostly, in the winter, and December, January would be the main amount of spores, and that's when in California gets most of the rain. So there's a correlation with the rain and the spore release here. So high spore abundance from late fall to midwinter. So pruning wound protection is essential during this time. During these months there is a mass of pruning wound protection, but also in California we saw that late pruning or delayed pruning can also help, because if we are able, to delay pruning, by the end of February, early March in California, we are out of the window of the highest amount of spores in the environment. So just by delayed pruning, we were able to show a significant reduction of infection. I just wanted to show you how we use this disease epidemiology and this data to try to find, target periods, target timing for conducting these practices. So here in BC is very different, because what happens if we do late pruning? Well, it's not going to work, because most of our, spores are going to start coming during the end of March, April, May. So if we are delaying pruning, we are actually making a boon when most of our spores are going to be in the environment, and that boon is going to be highly susceptible. So basically, late pruning is not an option for us here in BC. What we know is, if we are conducting pruning, during this time, we need to protect these pruning wounds. Now, finally, we have a chemical product we can protect our pruning wounds register, and hopefully we will soon see some more alternatives with some other biologicals or chemicals registered. But late pruning is going to work, and we are going to need to really treat these pruning wounds, during this time, because here in BC, most of the pruning is done, March, the month of March, early April. So what about early pruning? We have shown that, there is basically no spores in the environment here in BC during the winter. So what happens if we prune in December, January, or February and have more time for that pruning wound to heal? So when the spores come in March, April, or May, that pruning wound is less susceptible. So we conducted studies here in two different varieties, Merlot and Chardonnay. So here is the time of the pruning. We prune at different months, December, January, February, March, and April. And then we came back, when we know that the spores are present in the environment, and inoculate those pruning wounds. So here is the mean percentage of infection. So what we are seeing here is if we prune early in BC, December, January, we see a significant reduction of infection, which can go almost up to 40-50%. If we prune later, February, March, or even in April, those pruning wounds are going to be highly susceptible. The pruning wound doesn't have enough time to heal because those spores are going to be immediately present in our vineyard. So we showed that here in BC, early pruning can mitigate. It's not going to control absolutely 100%, but we can see a significant reduction of infection in our pruning wounds. And the other cultural practice, and this is the last part of my talk, is remedial surgery. So as I mentioned before, there is no products available to eliminate or to stop this vascular infection. One is inside. So the only option is remedial surgery, trunk renewal, or as many growers call the chainsaw solution. So we have the infection through our cordons. So basically we start cutting until basically we see no infection, and we are able to retrain the vine from this part. Advantages, well, we can, retrain the same clone and the same fruit quality that we have, in some old vineyards. Maybe that clone or that variety is not even available. We already have a very well-established root system, so those canes that we are going to retrain, they're going to be basically growing within a growing season and be back in production very, very early. The disadvantage is costly, it's labor intensive, and always, of course, the better results are done if it's done during the dry season period, because we know that there is a correlation with the spore release and precipitation. But nobody usually does this work during the summer, so that's one of the disadvantages as well. So what happened here in BC, as I saw you before, we're starting to see vineyards, collapsing with a heavy amount of infection. So this situation is in a grower. I was called, and basically I started to see in the last 5 years, the production was significantly going down. It was not getting the yields that usually it was expecting. So we start cutting the vines and we see, yes, there is, infection at the bottom. So the grower at this point have the solution of replant the entire vineyard, which is significantly costly, as you can imagine, and wait another 3-4 years to start in production, mostly because the infection was already at the bottom. So our hypothesis here is, okay, yes, there is canker at the bottom of the trunk. These vines were self-rooted, but let's check how long these vines can still be in production, even with the canker at the bottom, with the idea that if we can keep those vines in production, the grower will have time to start looking into replanting sections, little by little, while still having a cash coming into production. So these are the results for the Pinot Gris. These vines were cut in winter of 2017. So we see that in blue, those are the control, the uncut vines. In red are the cut vines. So these vines were cut in January, February of 2017. We already got about 1 kilo of fruit in per vine, in those cut vines. In 2018, we see that the cut vines were already no significant difference in production with the uncut vines. We can see how the controls, the production is declining. 2 years after, we already have higher production in the renew vines versus the uncut vines. 2020, 2021, these years we have significantly issues with the heat dome. We have a very poor shed due to spring rains, but still we see a higher production in the cut vines. In 2022, at the end of the experiment, the production in the renew vines was almost double than the uncut vines. So quite significant. All the vines were survived. It was 100% survival of these vines, and they were still in production after 6 years. We observed no difference at all with the fruit quality in Brix, pruning weights, pH, TH. So the new retrained vines, the fruit quality was exactly the same as the old mature vines that were left uncut. And at the end of the experiment, so the grower, when we started doing this work in the Pinot Gris, was getting about 7.5 tons per hectare. At the end of the experiment, we increased by 60%, 13 tons per hectare doing this work. In the Pinot Noir, it was about 5 tons per hectare. By the end of remedial surgery, 160% increase, 12 tons per hectare. So we can see that the vines were still alive, were survived, and we significantly increased production by conducting this work. And this is just a snapshot of how the uncut vines, you can see even the color of the leaves, the poor vigor compared to some of the vines in the back, very, very low fruit, and how, the grower left here in this case, 2 trunks, the amount of fruit we were able to get. So as a summary, we have, yes, the first chemical product registered in Canada right now to combat trunk diseases. We have identified very highly efficient biocontrol agents. So we hopefully are working towards a product formulation with locally found Canadian isolates for biocontrol. We have several cultural practices, like early pruning in BC, the pruning time of remedial surgery that we know it works, and we can significantly reduce and actually expand the lifespan of the vineyards. So sustainable control is possible. It's a multidisciplinary approach. Not only one thing is going to work. I think we have to work into implementing many of the options we have. And finally, I can say that Canadian growers, start having some solutions to mitigate this big problem in vineyards. So just thank you very much, to my team. None of this would have been, possible without all my team in Summerland, the Viticulture group, all our funding partners, as well as the University of British Columbia, Dr. Miranda Hart, which is a great collaborator of mine, taking on PhD and master students. I just want to show you, this is a review paper we published in 2018. A lot or most of the information you need to know about trunk disease is here. It's free to download. You can go to the website here up in Plant Disease Journal, and it's a very good overview of everything we have been talking today. And with that, I've done, and I just thank you very much for the invitation and the opportunity, and I believe we have some time for a few questions, so I will be happy to answer. So thank you.
Okay, so we have, perfect. Thank you very much, Dr. Torres. Okay, so we have 3 questions. I'll start with the first one. Have there been any trials on the impact of rootstocks against GDT?
On rootstocks, if I understand the question, maybe how the rootstock is going to impact the scion growth and the scion susceptibility of trunk diseases. I don't think, not to my knowledge, for that aspect. I know there have been trials looking into the susceptibility of different rootstocks to some of the jam vine decline. And there have been some trials on how, for example, Phaemonella chlamydospora causing Petri disease impact the rooting and the callusing, of production of those rootstocks. And we have seen that it can have a significant impact, a high amount of infection. In nurseries, they have a much higher issue trying to really produce, rooting and callusing those rootstocks. But in the sense of how the rootstock impact, the scion, not to the best of my knowledge. We have to think that these fungi enter through pruning wounds, and we are pruning every single year, no matter what, so there is not so much influence on the vigor that the plant may have, but it's basically, the wounding we do.
Okay. Any trials comparing GTD in vinifera versus hybrids?
No, and I would love to do some of that. So hopefully I can connect with my colleagues in Nova Scotia and Campbell. We know that hybrids are also susceptible, and there have been some work done in the United States, in Northeast United States, where they show that not only hybrids, but some of the wild, Vitis labrusca, riparia, they are actually susceptible to trunk diseases. But there hasn't been really proper, I believe that because we know that the vascular system of the morphology of the vascular system, the silent vessels, is very different depending the cultivar. We have some cultivars with a very wide xylem vessels. We have some cultivars with a very narrow and thin xylem vessels. So actually that is known. There are studies that can impact how the fungus is able to colonize and travel through the vascular system. So on top of my head, I'm not aware how this morphology on the vascular system can be between hybrids and vinifera. But knowing that there's difference even within vinifera, I suspect that there's a big difference, and that can play a role. So that's where the hypothesis I have that can influence having a hybrid, or even within hybrids, different varieties of hybrids can implement. But no, I think we have some opportunities here to conduct some work, yeah, with my colleagues at Kenville. So that would be good.
Okay, we have one more. What is the best way to scout our vineyards for GTD? What time of year is easiest to spot GTDs?
That's a great question, because as I mentioned before, the main problem we have with these diseases is we don't see the symptom immediately after infection, like we can see with botrytis or powdery mildew. So the spore is going to infect the pruning wound, it's going to start colonizing. We may not see symptoms up to 3 or 4 years later, which is already too late because we already have the pathogen inside. So we have specific diseases that, like Eutypa dieback or ESCA, the symptoms are very, very characteristic. Eutypa dieback is causing very short internodes. The leaves start capping, with some necrotic. There are very nice pictures in that review paper that you can look. So during early spring, early season, you can really check for those symptoms. Because the thing is, you can have one spur or one cordon or one cane showing those symptoms, but the other part of the vine is totally fine. It's depending where this fungi is. So once we go into the growing season and the whole foliage start taking over the vine, it's going to be harder to see. ESCA has very characteristic symptoms we call tiger patterns on ESCA, and you can see the leaves basically during the peaks of heat, and water needs for the plant. They start scorching, they have some intervenial reddening in reds, chlorosis in whites. So those are actually, and sometimes they suddenly collapse. So those are, but also it's common to see, as I saw, the canker might be inside the plant and we don't see symptoms. And this is typical with Botrytis, for example. Some of the things that growers can do is look for all pruning wounds. You have a cordon, for example, if you are cordon spur prune, look for all pruning wounds and see, sometimes they have flat areas on the bud because the canker start forming. So you see this flattening of the bud. That's a sign that you may have a canker running. Sometimes if you start seeing positions in your vine that they don't grow in the spring, the buds are not pushing, maybe the canker has already collapsed that spur, so that's another idea. But it's not always easy because many times we don't see symptoms, but the infection is inside. But there are different ways, mostly early during early season for some diseases, and during the highest peak of heat or water needs for other diseases when we can start monitoring.
Okay. We have a good discussion here today. So we've got one, let me see. How many applications of the product, and what is the timing for Senator?
So I believe the number of application, I think is they got me here. I should have read the label better, but I think it's 2. But again, thiophanate we have also to take into consideration that it can be used also for other things like powdery mildew, and it can create resistance. So I wouldn't advise, because it's not only for plant diseases. I wouldn't advise to do more than a maximum of 2, maybe just 1 application. It usually apply in dormancy. So you prune, and as soon as you have pruned, as soon as you can enter in that block and apply the product, is going to have this 14 to 21 days protection of these pruning wounds. So that's why I mentioned it's very important to know, if we look, for example, in Nova Scotia, I know we don't have data, but it would be very important to know when those spores are really happening in Nova Scotia related to your pruning time. I know your summers are more wet and rainy than ours, so you may have, a longer duration of spores during the growing season at a higher level. So maybe not so much. When is the main time Nova Scotia to prune? It's also April, March, April.
So most people have just finished pruning or are finishing up.
Okay, yeah.
And yeah, so tying's kind of midway.
Yeah, so I would say you just finishing pruning right now, and start warming up, and you probably start having rains coming. That would be the timing, because there's one thing in the label. I think in the label they include in the label to avoid application after bud break. I believe that's in the label, but yeah, it's a product that usually works for dormancy to protect this pruning on dormancy.
So let's see. Do we agree that regardless of all factors involved, trunk renewal is potentially the best solution?
I would agree. Yeah, I could agree if we can do it. Just to give you an example, Australians are masters of trunk renewal, and most of the work we know in trunk renewal comes from Australia. My colleague Mark Sosnowski, pathologist there, has done incredible work on trunk renewal. And it gets to a point that is a factor implemented in the production system. Many growers in Australia, they know when their vineyards get to 15, 20, 25 years, they are going to do that work to keep that vineyard functioning. They don't even bother to start looking where the canker is, and start looking, if I only cut the cordons and retrain, or if I, they just go all the way down, cut, because they know that. They know, depending the variety of where you are, depending the pathogen, year 15, year 20, the canker has make it all the way down. So before they do that, they cut and retrain, and they keep going with production. So yes, I would agree. There are some difficulties with trunk renewal. Is much easier to do if you are self-rooted, definitely, because you're going to have way more suckers coming, out of that. It's something that maybe Nova Scotia is more difficult, because the phylloxera issue you have in Nova Scotia, and most of your vines, I believe, are grafted. But I might be wrong, but I don't know. My
The
understanding
hybrids less so than
Yeah.
the vinifera. So yeah.
So, but once you have a graft, if your vine is grafted, you are more limited, because of course you can only take suckers, above the graft union, and that's talking about the rootstocks before. Depending the rootstock you have, or you are gonna have more or less developing of size. There are some rootstocks that really limit, the production of suckers. Others, they are more vigorous. So that's a problem. But yes, I would agree. The data we have here and we've shown is even an infected vineyard where we knew the canker was at the bottom. We already got, and the vines are still producing. We already got, 8 years of production, which eventually these vines will die because the infection is at the bottom. But the grower now has had 8 years of cash coming in, and is actually what the grower was doing is every year 5 to 8% of the block was being replaced. So slowly, keeping the economically viable the production. So yeah, I would agree. Yeah, it's a good practice.
Are there plans for nurseries to test their own propagation materials?
So that depends a lot, and depends on the nursery and depends on the production. I know nurseries that they have an implementation of testing for trunk diseases at the nursery level. There are other nurseries that they don't test. So it's going to depend on nurseries. We have to think in one thing. These fungi are not regulated. So, and not only in Canada, they are not regulated anywhere in the world. There is no agency, that regulates these pathogens as quarantine pathogens, because we see how ubiquitous they are. They are almost everywhere, and they are not only on grapes. They are in many hosts, even in native plants. So of course they are not regulated. That makes more difficult, to implement, sanitary, to the nurseries. If we talk about viruses, okay, we have viruses, they are regulated. We have phytoplasmas, they are regulated. We have ways to eliminate, the viruses, or to produce clean plant, virus-free. It's very difficult with these fungi. So, but yes, there are, like in every, there are better nurseries than others that are doing everything possible to mitigate the impact and infection. And you see also in the planting material, even in some of the nurseries we have been testing, we see nurseries with a much lower amount, and then you go back to the nursery and they tell you, yeah, no, we do this, we do that, we do this. One thing that have changed in the nursery in the last 10, 15 years is they treat the mother blocks same as it would be a commercial, and before it was not like that. Before usually, you get your powdery mildew because you are not producing fruit. So, and one of the things that I see changing a lot is they start really protecting these pruning gums. Mostly, you think a mother block of rootstock and those big plants with so many cats, where they collect all the rootstock, they really start working on trying to protecting those plants. So this has changed. So hopefully, little by little, we start seeing improvement. But no, it will be depending on the nursery.
Okay, thank you.
As your nursery, when you get in material, what they do, in that, it's a good thing to ask.
Okay, I think if anyone has any last questions, please put them in the chat right now. Otherwise, we're going to be closing up pretty soon. Awesome. Has there been any studies done on, so we've got heat stress, but is there anything being done about actual like cold weather, climate, and?
So we have tried. Actually, we have one experiment where we wanted to test, the effect of cold in these diseases. So very similar experiments. We have dormant canes artificially inoculated. Here in Summerland, same as in Kenville, we also have these specific freezers that will do bad hardiness, and we can ramp those freezers, to very low temperatures. So we have tried. The problem we have with these cold experiments is when we use potted vines, we start getting, to temperatures that we wanted to test, -10, -15, etc. We are getting actually more damage root, the root damage, the roots in those pots, they really get completely hammered. So we were unsuccessful. We are trying to develop, a methodology that we can do first in the lab. But ideally, we would do in the field, but as when you plan experiments in the field, you look for cold, and maybe you have 10 years with no cold, hopefully. But you look for heat stress, and then you have 10 years of summer where there is no heat stress. But so it's very unpredictable. So we are working to develop a system that we can really test that first in vitro in the lab. But we see a correlation with the cold. For example, here in BC, the previous cold damage was in 2009. We have a very strong winter freeze in Thanksgiving. Canadian Thanksgiving, we went to -18. The vines were still not shut down here. There was still a lot of water in those vines. So what we observe is big cracks. The vines split, even at the cordon level, trunks, and we have seen a lot of those vines, move forward on with a much higher amount of trunk disease. Why? Because now we have a wound, a split all the way in the trunk, all the spores get in there and basically finishing the vine much quicker. So there is some correlation with the effect of winter can have with these diseases. What I would love to try and to prove is winter conditions in Canada are favoring these diseases to progress faster than other, because we grow grapes and the winters are still pretty cold no matter where you are in Canada. So that's something that we have, we are planning to do, and hopefully we can figure out how to do it.
Awesome. Thank you very much. I think this has been an excellent discussion. Thank you everyone for joining us today. If you have any further questions, email them to me and I will forward them on. And again, thank you very much for joining us today, Dr. Torres, and hopefully we'll see you all soon. Yeah,
thank you very much for the invitation, and all the best for this growing season. Hopefully you get everything you're looking for. Okay?
Thank you. See you soon. Bye.